Wait, over here.
Oh, sorry,
Okay.
sorry.
So, phospholipids make up the inner membrane in the bilayers. So basically what this does, it allows only for non-polar molecules to cross the membrane without the help of proteins. The reason for this is because phospholipids are made up of polar heads and non-polar tails. So basically, if there's a polar molecule that wants to cross the membrane, it's going to need the help of a transport protein, such as the carrier protein right here and the channel protein right here. The way the channel protein works is by having a channel in between the protein, basically allowing for non-polar molecules to cross the channel into the membrane. And the way the carrier protein works is similar to the transport protein. The only difference is that it's more specific. So basically what it does, it allows for several polar molecules to attach to the protein. It'll change in conformation of the shape, and then it'll shuttle the molecule across the membrane, such as shown in the picture. And this type of diffusion through the membrane is called facilitated diffusion, because it is needing the help of a protein. Any type of transportation across the membrane that needs the aid of a protein will be called facilitated diffusion. And anything that does not need the help of a protein to cross the membrane is called simple diffusion. So basically, osmosis is a type of simple diffusion. The way it works is from high concentration gradient, it's going to go down its gradient, so it's basically going from high concentration to low concentration across the membrane. So basically how it works is really similar to osmosis, and basically, wait, what?
Sorry, the tower is just so beautiful.
So, okay, so basically, hypertonic is just basically when the concentration of solute is greater outside the cell than what it is inside the cell. So an example of this would be when an animal cell that does not have a cell wall gets into a hypertonic solution, the water is going to want to go out of the cell, making it shriveled, and it's not good for cells because it'll die. And then hypotonic solution is when the concentration of the solute is greater inside the cell than it is outside the cell. An example of this is when an animal cell without a cell wall is inserted into a hypotonic solution. The water will want to go in. It's going to burst and it's going to explode. This is called lyse. But if a plant cell is placed into a hypotonic solution, the plant cell, since it does have a cell wall, it's going to be turgid. It's going to be healthy. It's going to be good. It's a good thing for a plant cell to be in. So basically what happens with, oh, I think I wrote this wrong. Yeah, water goes.
Come on,
into
man.
the cell wall, man. Okay, so basically, if a plant cell with a cell wall is inserted into a hypertonic solution, it's going to be plasmolized, and basically the water just leaves, such as it does in the animal cell, but it's not healthy for any of them either way. So it's just gonna like kind of shrivel up, but it's called plasmolized.
Plasmolized, yes.
Yeah. So for an isotonic cell, it's basically, well, a cell inside an isotonic solution, it's gonna be flaccid if it's in the plant wall. I mean, if it's in the plant cell, because they have a cell wall, and it's just gonna not be good for the cell. Basically what isotonic solution is when a concentration of the solute inside the cell is the same as the concentration of the solute outside the cell. While this is not healthy for plants because of the cell wall of the plant cell, it is healthy for animal cells because they do not have a cell wall. So therefore, osmoregulation is very important in animal cells because they do not have a cell wall. Okay, I think we're good.
Okay, and this is why it is important to know the structure of the membrane.